Hello, everyone. Welcome to Pint of Science 2021. We've got two very exciting talks for you this evening. So, Pint of Science runs every year in May all across the world. We aim to bring scientists into accessible venues to communicate their work to the public. Before we start, there's a chance to win some Pint of Science merch. All you have to do is fill out the feedback form linked in the description, and the competition closes at the end of June. First off, we have a talk from Newcastle NIHR MIC. They evaluate diagnostic tests to ensure the most effective tests are delivered quickly to the UK public. We're joined by Kyle and Tim this evening, who are going to tell us about their work evaluating COVID tests.
Hi there. Thank you very much for that. So yeah, we're going to talk today a little bit about our work and COVID testing, and particularly what makes a good test. So to start off with, who are we? Well, we're the Newcastle MIC, which is the MedTech in Vitro Diagnostic Co-operative. We're one of several MICs working under the National Institute for Health Research. So the MICs were created to help companies, clinicians, and charities make sure they're generating good, robust data so that they can bring new clinical tests into the NHS. So the work that we do can range from data analysis and statistical modeling all the way to interviewing clinicians and patients to get their input on how illnesses are diagnosed, managed, and treated. So Tim and I are known as mixed methodologists, as we both do computational analysis and face-to-face, or in this case, Zoom-to-Zoom investigations usually. Here at Newcastle, our team is split between the university and the hospital trust, and that gives us like a really great access to researchers, medics, and patients. And we also work really hard to involve members of the public in our work. So we have like a wonderful group of members of the public who really sort of help tailor our work and maybe do some sort of lay summaries and things like that that really help our work have a greater audience. So a lot of our work at the moment on COVID, is falling under a big national group called Condor, which is the COVID National Diagnostic Research and Evaluation Platform. And some of the work that we'll talk about today will be based underneath the work that we've done for Condor. So just as a disclaimer, anything that we talk about today is our own views and not necessarily related to the NIHR or the Department of Health and Social Care. So we do work with quite a few different types of tests, and I've just shown a few here. So if you're working, for example, with a screening test, that would be most likely to find a disease that's in an asymptomatic population. An example of this will be like breast cancer screening. Diagnostic tests are one that we work with quite frequently. These usually are to decide if a patient has a specific condition or not. And this might be like usually a yes or no, for example, like testing for anemia. We also work occasionally with prognostics. These are more to determine what an outcome is likely going to be for an individual patient. So this could be like tumor grading in cancers is quite a common one. And there's also monitoring tests, and these are a lot more about how a patient is changing over time. So an example of this might be someone monitoring themselves for diabetes through glucose monitoring. Today, most of our talk is going to be about diagnostics, because that's pretty much what we're doing with COVID. But before we get into that, a little primer, I think, on some of the work that we do and the information that goes in behind the scenes, I think, for some of our projects. So the table that's on the screen now, this is a table that we use quite a lot in our work. It's called a 2x2 table for obvious reasons. And we use this to determine how well a test is performing. So on this table, we can work out the accuracy of a test, which is its combination of sensitivity and specificity. And we can see at the side there, the sensitivity is how often a test correctly identifies someone who has a condition. And the specificity is how often a test correctly identifies somebody who does not have the condition. So looking at the four quadrants here, a diseased and positive patient, or someone falling into this bracket under disease and testing positive, would be a true positive. If they were diseased and tested negative, then that would be a false positive. if they were healthy and tested positive, that would be a false positive. And if they were healthy and tested negative, that would be a true negative. So in an ideal world, we'd have a perfect test, in which case every single person that was tested with the test had a correct answer. And so all of the patients that you tested that had a disease would be positive, and all the patients that were tested and didn't have a disease would be negative. And in this case, we'd see that there'd be 100% sensitivity and 100% specificity. But in reality, this is rarely the case, and tests are usually never 100% accurate. So what we end up with is a few false negatives and a few false positives, and these will then affect the sensitivity and specificity. So in this case, we've got 90% specificity because 1 of our 10 tested people with a disease has tested negative when they should have been positive. And we've got a specificity of 80% because of our 10 healthy people, 2 of those tested positive when they should have tested negative. So I'll hand over now to Tim, who'll talk you a bit more through the COVID side of things.
Hi everyone, and thanks Carl for that introduction to diagnostic tests and the accuracy, so sensitivity and specificity. I'm going to kind of complicate things a bit more, and COVID is a great example to use. I know we're all sick of it, but it kind of drills down into what's important with these tests. So COVID diagnostics can tend to be thought of in 2 categories. They're either lateral flow tests, which you do at home, which are dead cheap, take less than an hour to get a result back, and real-time PCR, which are a bit more expensive, take about a day to get the result back, but they often get sent to the lab and need a specialist to interpret them. Now, these tests differ in the sensitivity and specificity as well, with lateral flow often being a lot less sensitive, a lot less specific than PCR tests. It's kind of the balance you get with cost and user. But to complicate things a bit more, I'm now going to talk about how the performance of tests can change based on the setting alone. So for this example, we'll take the test Carl talked about before with a 90% sensitivity and an 80% specificity. And we'll say to test for COVID that you're going to use in the emergency department for people coming in with symptoms. So rough figures say 10% of the people coming into your emergency department have COVID, and you're applying this test to them to try and capture those with COVID and those without COVID. This could be to diagnose them, it could be for infection control, whatever. So with a test like this, so 90% sensitive, 80% specific, which is pretty good performance for a test, out of those testing positive, you would get 9 out of the 10 of them. So quite a high hit rate. But you would also get another 18 testing positive who are actually negative. So this is where you can think of the false positives. But this is with symptomatic patients. If we then look at, say, asymptomatic patients in a school or a mass screening, so just in the workplace, people who don't have symptoms are probably less likely to have COVID, and then say we have 1%. So with the exact same test applied in that setting, sure, you pick up the person who's positive, but you also pick up another 20 people who aren't positive for COVID, but the test reads them as positive. So you can see how change in the setting of the same test can hugely change the probability that your test result will come back positive or negative, which is quite an issue when you're rolling out these tests. You do also get all the negatives correctly, but we then need to start thinking about kind of the implications of testing and what else could factor in. Because remember, in this scenario, we have only changed where the test is used, not what the test is, not who's used it. It's purely where it's used in the population there. So next slide, please. So some of the other things we need to consider with testing is who should be tested. Should we test purely the symptomatics because they have a better chance of being positive, so we'll have less false positives? Should we test the asymptomatic to try and enable them to go back to work, or those without symptoms, I should say, not asymptomatic, to enable them to go back to work and have a confirmed negative? And we need to focus on kind of what the prevalence of the disease is nationally and locally, because it's going to change across different centres. So this kind of boils down to what test is best for what situation. So if you want to diagnose someone as COVID positive to help with their treatment, you would ideally be looking at something like a PCR test, which is going to pick up the majority of people who have COVID, because that's what you're focused on. But this also boils down to who should perform the test as well. So there's a study on the slide there done and published in Nature, which showed that with the same test, the rapid antigen test, which is similar to the lateral flow devices, the sensitivity of the test changed based purely on the user of the test, with laboratory scientists getting a sensitivity of about 80%, where the same test, members of the public got it to be about 58%. So you can see how multiple factors start impacting an accuracy of a test. So you can have a test that's rated highly, like ours, 90%, 80%, but then when you actually implement it in a real world, that performance can sharply decline. And thinking away from the test as well, you need to think about the consequences of a false result. So for those asymptomatic testing in schools that I just mentioned, every one of those false positive tests would have to isolate at home, their families would have to isolate, their classes would have to isolate, and that ends up building a huge burden on the individual, not just on the healthcare sector, but on the individual. It'll be a mental strain for them, it'll be financial considerations needed to take into account, absence of key workers, so absence of the teacher will impact other students. So we start to need to think about these before we implement the test. And one of the main consequences is if you have a test, so-called a bad test, as we're talking about good tests, that people then start to trust in the result, you undermine all the testing effort. So if you have a test that has a high proportion of either false negatives or false positives, you can't trust the test. You will end up with people ignoring it. They will take a test, say, I feel fine, the test must be wrong. And we can't have that. So next slide, please. So kind of sum it up, tests are rarely perfect. Even with, say, pregnancy test, you will still have false negatives for a pregnancy test or false positives. You never get a test which is 100%. But even lower accuracy tests, such as like lateral flow devices, can be useful in some circumstances. So the key thing with the lateral flow device is we got that one person out of 100 who was COVID positive. But if we followed it up with an additional PCR, we would then reduce those false positives and enable people to go back to the classroom. So using it in a kind of testing regime might be the better way to use it. Whereas a bad test used in isolation can be worse than no test at all. You can either end up with too many false positives or too many false negatives, which will increase the chance of spreading the disease further, or it can just undermine tests entirely. So how can we improve testing in the future? So the best way to do it is to try and understand the system that the test can be placed in. It's to run your tests and build the evidence with the correct users. There's no point designing a test for use, say, in a care home, and then only having laboratory scientists test the use. Your figures are going to be way off and it might not work as you expect it. And this is kind of where the MIC comes in. A lot of our work is based around understanding those pathways, understanding where the test is going to be placed, looking at the accuracy, evaluating it in conjunction with the benefits and harms for the individual, and really trying to boil it down into all these factors and see where it would have the highest benefit with the lowest risk. So I think that quite sums it up. So if anyone has any questions, I don't know if any have been posted in the chat already, but me and Carl would be happy to answer them. And you can always contact us through our website, our Twitter handle, which is on screen now, and we've just started a newsletter that will be doing its round soon. Thank you.
Hi, hi. Thanks so much for those talks, Kyle and Tim. They're really interesting. So yeah, just to get us started off, could you explain a little bit about what the difference between lateral flow and PCR tests are, and kind of specifically a little bit about PCR tests would be interesting?
Yeah, so PCR tests tend to look at the genetic material. So they find the smallest piece of genetic material and amplify it up. So you run it through reaction and it builds and builds and builds it. And you can pick up COVID early on in the infection cycle. Downside is you can also pick it up later on. So it's kind of balancing the understanding of the test, which is why they're interpreted by laboratory scientists. You need that kind of expertise to boil it down and understand it. Whereas lateral flow tests, they detect kind of an antigen that's from the swab. So it's often people say they detect the like infectious COVID or infective COVID, which the data is a bit, it's not quite there yet with whether that's the case or like what time frame. But with lateral flow tests, you tend to detect those with a high amount of COVID in the system. So your time window is a lot narrower with the test. So it's two similar tests in a way, but completely different systems and how you have to use them.
Okay, cool. And yeah, Becky Jones has a question. How do the different variants affect the accuracy of the tests, or or is it kind of fairly consistent between the different variants?
I can take that one if you want.
Yeah.
Yes, so yeah, there are a number of different variants, and obviously some of these will probably have more of a different effect. So as Tim was saying with the PCR tests, if they're picking up a very particular part of the COVID sort of genetic material, if that has changed enough that it doesn't pick it up anymore, then the test won't work for that particular variant. I think in the most cases, it's generally quite fine because there is a little bit of leeway on this, and actually there's usually multiple regions that are tested at the same time. So usually the variant shouldn't have much of an effect. But as these are being generated, we've seen now with the India variant and more other variants coming up, clearly these haven't actually been tested in a clinical setting because it didn't exist at the time when the test was first being brought out. So the effect that some of these might have on the tests is probably unknown yet, or the information on that's probably still being developed.
And can I just add to that, actually? So you might hear in the news about genetic sequencing as well, and that's where they've identified one of these strains and they're trying to understand the genetic material within it, so then they can then adapt the tests and develop new tests, which will then pick it up.
Okay, yeah. So yeah, is that the thing where they can kind of sequence all the DNA and then send it off to everyone around the globe, so that everybody's kind of using the same copy, as it were, of the data?
Yeah.
So yeah, and Rachel M says, given all the data, what and kind of everything you've said, what are your opinions of the recommended twice weekly testing rollout in the UK? And do we need training for people doing this at home really to increase the accuracy?
Do you want me to take this, Carl?
Go for it. Yep.
Yeah. Right. So yeah, ideally the better trained person, the better, obviously. But the way lateral flow devices are being rolled out, there is benefits to rolling them out. You will catch them. You will catch a positive case. It's just how many false positives you catch and how kind of what the resource cost would be to train everyone, because even with frontline healthcare staff, the swab can be, it's about 70% sensitive, the PCR swab. From the data I've seen, 70% sensitive. Might have changed since people have learned how to do it. But yeah, training would be ideal, but it's one of them things you need to balance up availability of the test and training. And that's kind of where the evaluations come in. So we'd look at usability of the test as well. So we'd get a few tests, send them to groups of volunteers, essentially groups of one of the ones they did was school teachers, I think, and to see how the sensitivity impacts that. And by looking at that change, you can kind of gauge whether you need to roll out training. So if say they did an LFD trial and it came back as 20% sensitive, you wouldn't go with that test unless you had training. So it did limit who could use that test. I hope that answers the question without.
Yeah, it was very interesting, certainly, and it really gives you an idea of kind of how many things there are that need to be monitored with all of this, kind of beyond just the basics of the accuracy of the test itself, kind of how much data there must be to monitor. And Rachel M as well there was asking, do you have any tips for trying to make sure that tests that are completed at home are safe and kind of are completed as accurately as possible?
That's tricky anyway, because I've never performed a test myself, but it's just follow the instructions as best you can. If in doubt and you're symptomatic, I'd say still book into the regional test centres, because it's going to be more accurate, basically. It'll take 24 hours or so to get the result back, but if you're concerned about the accuracy, I'd definitely go down that route as opposed to using an LFD test.
Okay, cool. So yeah, I don't know if there's going to be any more questions to come. It doesn't look like it. So yeah, thank you both again for the talk. It was really interesting. Yeah, kind of everything was really informative as well. I feel like I've got a much better understanding of what's going on from that. So yeah, that was really good. And thank you to the audience for offering up questions there as well.
Thanks a lot.
Thank you very much.
So next up, we have Ricardo, who's going to talk to us about his work on tissue engineering. Ricardo's worked on a lot of exciting projects, including one developing 3D printed corneas right here in Newcastle. And today, Ricardo is going to talk to us about his current work with lab-grown meat. So, hi, Ricardo.
Hello, Alex. Hello, Jack. Hello, everybody. Thank you. Well, first and foremost, let me thank the organizers of Pint of Science for the kind invitation to give this talk today. My talk is entitled Out of the Lab into the Frying Pan, or the Science Behind Lab-Grown Meat. My name is Ricardo Gouveia, and I'm a scientist and entrepreneur based at Newcastle upon Tyne. And in the next 15 minutes, I'll be introducing you to this exciting new field of research, explaining what it is, why it's important, and how it's done. Now, with the invention of agriculture over 10,000 years ago, humanity started its journey to become the dominant species on planet Earth. Agriculture gave humans a greater degree of control over nature in terms of nutrition and food security, and was fundamental for the development of large and densely populated societies. Well, with these traditional agricultural practices, animals had an extremely important role and served not only as a source of food and leather and other materials, but also they helped farmers with heavy labor and managing the land and managing crops. However, since the Industrial Revolution in the 18th century, agriculture became more mechanized, more automated, more efficient. And so animals became less and less used as labor within our food production systems and started to be mostly regarded as foodstuff. In particular, animals became the prime, most desired source of proteins, with a demand that in time led to industrial-scale animal farming we have nowadays. But still, this is not enough. And in searching for producing food systems more efficiently and more independent from nature, we are now exploring the next logical step, which is lab-grown meat. This is the production of animal-based foods, but without using animals, taking advantage of sophisticated bioengineering technologies to biomanufacture what we need without having to raise or even slaughter animals. You will definitely say, surely this is only science fiction. But is it? Indeed, it is not. Supported by multiple scientific and technological advances from research areas such as cell culture, and biotechnology, and bioengineering, as well as cellular and molecular and chemical sciences, as well as synthetic biology and biofabrication, even animal husbandry. lab-grown meat can now be actually a reality. Frequently, these advances they came from a medical field or a biotechnological field. And typically they were advances that were attempts to find a cause and the cure for human diseases. However, now these same technologies can be applied to the production of food. And it's very important that we do so, because lab-grown meat can help prevent or at least reduce the impact that mankind is having all around the planet. For example, by reducing the damage of human activities on the environment, including deforestation, greenhouse gas emissions, or overstock depletion. By improving animal welfare or reducing animal poaching, by preventing the appearances of new diseases, and also by reducing food insecurity and poverty globally. Because the truth is that the global population continues to grow, and so does the appetite for meat. And this is despite the limited natural resources that are increasingly overstrained. So lab-grown meat can help us prevent such depletion and achieve a greater sustainability. Now, you may say, this is a radical solution. Surely we can try other things, like convince more people to stop eating animal meat. But to that, I reply, yes, but we already tried that. And for one reason or another, people still eat meat. So we think that it takes a disruptive technology to find such a solution. And this disruptive technology can also bring other unforeseen benefits, like developing new technologies for food production and biomanufacture, develop new ways to produce things, have new scientific findings coming to light, new medical discoveries. All of these things, very exciting prospects for future development. For now, lab-grown meat production is mostly based on technologies that allow the cultivation of biological tissues from stem cells. That is why it is also known as cellular or cultivated meat. Lab-grown meat is a product formed by animal cells, typically muscle and fat cells, that are isolated from animals and then grown using more or less sophisticated tissue culture techniques and biomanufacture methods. It basically starts with the isolation of cells from an animal biopsy or sample, which are then isolated and expanded using what we call aseptic techniques and fed with a nutrient-rich environment, liquid environment, that we call culture medium. Next, these cells are grown in larger volumes to form whole tissues that can then be harvested and processed to create what can be used as food. And the result is, at least theoretically, quicker, cleaner, and more efficient. Because contrary to meat obtained from traditional animal farming techniques, lab-grown meat can be produced without antibiotics, without any kind of pathogens, in a completely aseptic way, preventing the introduction of other contaminants into the food production system, and using less resources, less pollutants into the air, water, and soil. There are, however, many challenges to produce the huge amount of meat required to feed the world using our current technologies. For example, in the figure, you can see just a few of the flasks, cell culture flasks needed back in 2013 to produce one beef burger. So if you consider that on average, a person eats about 40 kilograms of meat per year, we would need billions and trillions of flasks to grow enough meat to fill our current demand. Enough flasks, actually, to go around the entire solar system twice. So the solution is, of course, to increase the volume and efficiency of the culture using, for example, larger containers, such as what we call bioreactors, or to increase production speed, for example, by using these new technologies involving 3D bioprinting at industrial scales. And so this leads us to here at Newcastle. Our Newcastle University Research Lab, it's located at the Centre for Life since 2014, is led by Professor Jay Common, who is a pioneer in biomedical engineering. Here we developed world-class research on how to create tissues for human transplantation, mainly by developing methods to control cells into forming new tissues, by designing new solutions to feed cells in culture without the need of animal-derived ingredients, and by artificially reproducing biological tissues with exactly the same structure and function of their natural counterparts, or as I like to call it, the real thing. All this allowed us to create human tissues with great precision. You can imagine it as relatively easy for us to convert our animal expertise into doing the same thing, but now with animal cells, and create lab-grown meats as a food source. Basically, also controlling cell growth, developing animal-free medium to feed these cells, and ultimately reproduce tissue structure and function of the real thing, including its taste. And so ultimately we became one of the first labs in the entire world doing research on lab-grown meat. Research supported both by governmental and non-governmental organizations. Research that also prompted us to create this Newcastle University spin-out company called 3D Biotissues. 3D BTO, as we call it, is a Newcastle-based biotech startup that is helping us to translate our inventions and discoveries in an academic background into the market. Our work here is allowing us to better realize what are the truly big challenges in the field of lab-grown meat, as well as to understand how and when can we solve them. In the past few years, we have perfected how to control cells isolated from animal muscle and use them to generate highly organized muscle tissues in a petri dish. We think that these discoveries will allow us to start producing lab-grown meat in small quantities in the beginning, but then later reproduce the real thing at scale in much larger quantities. And importantly, to reproduce it in a way that it becomes indistinguishable from natural meat and ultimately fits the demanding palate of we, the consumer. In the end, we aim at developing a solid scientific and industrial background that, combined with other innovations and social economic advances, will allow us to create a brand new food source that is, in a way, independent and not reliable on fluctuations of the natural environment. This is, of course, incredibly exciting. It's truly revolutionary. And this exciting prospect has definitely been demonstrated by the hundreds of people around the world that have now started to work and invest in large measure with their time and money into this area by supplying funds that are required for startups to do research in this area, but also at an academic level at universities, and even pushing governments and societies at large to rethink the very concept of food. So I think that the race to produce lab-grown meat affordably and at scale has already started. However, its final form might very well depend on the way it is produced. So you could envision it being made mainly locally within communities at relatively small scales, kind of copying the models of microbreweries that we see growing more and more around us. But alternatively, these might go into a multinational, massive production plant kind of model. Ultimately, I think it depends on us, the scientists, and on you, the public, to drive this forward and drive the final outcome of how this new kind of food is produced. So it is, in my view, very vital that we keep everyone informed and updated in the latest trends in this field. So that's it from me today. I thank you very much for your interest. If you want to know more about this, for me, fascinating new subject, I invite you to have a look into the many sources of information that are now available on lab-grown meat, mainly those supported by our incredible funders, the New Harvest Research Institute and the Good Food Institute, both American non-profit organizations that are driving state-of-the-art research and allowing the translation of state-of-the-art research into this area, or as well contacting me via the contacts I provide here below. So yeah, thanks very much for attending today, and feel free to post your questions online. I will try my best to answer them.
Thanks so much for that, Ricardo. It was really interesting. I'm a vegetarian, so lab-grown meat is a very interesting topic to me. And if you're watching at home, do remember you can post comments in the chat. So yeah, to get us started, Alex asks, do you think that lab-grown meat will eventually become more popular than plant-based alternatives?
And that is a good point. I think they will be complementary ultimately. This field has received a lot of interest in part because the plant-based meat alternatives started to become very appealing. It would be very difficult to predict which one will become the predominant food source. What I can say is that meat from animal is still very much a prestige food product all over the world. So even if you reproduce very finely the texture and taste of animal meat using plant-based products, there's still the social aspect of animal meat eating that is very much at play. So I ultimately think these things will be coexisting and complementary without a massive one overtaking massively the other. This is my expectation at the moment.
Yeah, so there's like a kind of cultural aspect to it. as well as kind of economic and science aspects as well,
Absolutely.
all happening at the same time. And Ella Louise Handley asks, realistically, how far away from lab-grown meat being produced on enough of a mass scale to provide enough for the whole of UK are we?
Yes, that is a fantastic question. We have seen many companies around the world promising a lot and constantly saying it's only 2 years from now, and it's only 1 year from now, only 3 years from now. But the amount of biomass produced needs to increase hundreds of fold in order for this product to become a staple food that is accessible and affordable to everyone. Currently, the technology really is not ready to produce all the tons required to feed a mass population. But this is definitely being looked at, and it has received a lot of investment from private investment from companies, as well as from governmental and non-governmental research institutes to look into this. So increased biomass production is a huge challenge, and is a challenge that is being addressed currently. So one way to sidestep this and to bring these products earlier to the consumer is to actually do a combination of animal derived meat produced in vitro with a filler or a scaffold, something that you can combine that is plant-based, so you don't need so many animal cells to create a product. And we foresee that as technology develops, the percentage within the product of animal cells increases and the percentage of filler will decrease. I believe that within the next few decades, we will have a product that is fully synthetic and is fully animal. Will be solely comprised of animal cells and other components that the animal cells produce to form the tissue.
Okay, yeah, that's really interesting. And Laura Score asks, does lab-grown meat contain the same vitamins and minerals as normal meat? Beef, for example, is full of iron, but the iron in beef has a different bioavailability than the iron in plants.
This is one of the beauties of having such a system, is that you can ultimately regulate the composition of the product of the meat. So in nature, we can source meats with different qualities and different compositions, different nutritional values, as also as different tastes and textures by selecting a different cut or a different animal or a different breed of animal. We can incorporate the differences found in this natural variety into the bioprocess in order to simulate exactly the same type of nutritional value or even an enriched nutritional value to these kind of products. So by having this ability, this plastic ability to regulate what kind of cells, what kind of percentage of fat cells or muscle cells, then we can regulate the nutritional value and the digestibility and texture and taste of these products seemingly without any kind of limitation.
Yeah. So, would it be then that we'd end up with kind of quite a lot of different products available with these slightly different balances as well?
Yes. So we very much believe in personalized food. So you could very much think of it as a healthier food source, because then you could have it designed to a specific human physiology. Let's imagine that someone requires more iron, as it was suggested. Maybe the meat product that you are eating or that you buy will have a greater iron availability. Maybe it is vitamin B12 that you require in greater quantities, and you might require, or carotenes, and you might then search the meat that fits your, not only your palate, but also your nutritional requirements as well.
Yeah, that's really cool. So Rachel M asks, how ethical or veggie friendly is the growing or culture medium? And she says that she thinks it contains animal byproducts.
Absolutely fantastic question. Currently, the majority of people that are growing cells for lab-grown meat, they still rely on a source of growth factors that is more easily procured in animal sera, so serum that is derived from animals. This is, of course, counterintuitive, because ultimately the final objective is to get rid of any animal derived product. So there is a huge focus from many groups, including ourselves, to find a source of alternative growth factors that can be used in a feeding medium without any kind of animal origin. This is already possible. These so-called serum-free media already exist. The problem is they were designed initially for biomedical research. So you can imagine that biomedical research is not as constrained by price, by cost, as food. So they exist, but they are quite expensive. So one of the drives is to find alternatives that are cheaper and more affordable to source in large quantities. But yes, that is a good point. But the point that is being actively researched, what we want in the end is to grow the cells with animal cells isolated from an animal specimen. So there is no, ethically it's very interesting because you don't need to hurt the animal at all. There is no need for slaughter. And the cells then will be grown in a feed medium that does not contain any animal product.
Okay, cool. Yeah, so there's still a bit of work to do, but kind of the eventual aim is for all of those mediums to be ethical.
Absolutely.
Yeah, and Hayden says, you mentioned a benefit of lab-grown meat is that it doesn't require the use of antibiotics, but would these still not be required to stop otherwise thriving bacteria in the actual culture, or like contaminating the culture?
If the culture is well made, you don't need antibiotics. This is a bit of a nerd point, I would say, but sometimes I like to say nerd things. But ultimately, antibiotics actually reduce the amount of growth in cells. So you don't want antibiotics in the culture. The interesting part is that if the culture is made in a completely aseptic, controlled environment, you do not need antibiotics in that culture, and that is easily done currently in many conditions, including in pharma, in the production of pharmaceuticals. So antibiotics normally in our lab, they are used to control contaminations, not to prevent them. If you have a good aseptic technique, then you do not need the use of antibiotics. In fact, it can be counterproductive.
Okay, cool. And Charlotte Huddart says, have you yourself tried lab-grown meat yet? Or if not, has anyone?
Yes, so I haven't. It is a matter of amount to be produced, so availability, let's say. However, in 2000, some people already have tasted it. There is even a company in Singapore that received, I think, late last year, approval from the Singaporean authorities to start selling a chicken nugget that is produced, that is cultivated. So there are people that tasted it. The first one, to my knowledge, the first one that was tasted was back in 2013, the one that I referred. It was a project from Professor Mark Post from Maastricht University, and he produced a burger that was then cooked and tasted in London by a chef and food critics. And that burger was a patty about 50 grams, and it cost $300,000 to produce. Currently, you are able to, the technology has evolved in these last 8 years that you can produce the same patty for a very small fraction of the price, about $50. And we are still trying to get the conditions to really lower the price to really affordable costs. There are many people that are able to spend $50 in one burger. So in order for this to be a source of food for the mass population, we have to have this produced at scale and at lower costs. Now, one thing that I will stress, definitely this culture meat has been very well monitored in terms of regulation. So I'm sure that before the general public starts tasting meat, the regulators will do so.
Cool. And Steve Ward asks, would we still have to keep animals in captivity in order to harvest the cells, or would we have some sort of cell agriculture?
It really depends, I think. I anticipate that we will have a combination of cell lines, so cells that are isolated for their ability to grow indefinitely, even without any kind of genetic modification. But I also anticipate that you will have animals that probably are maintained in much better conditions, in possibly traditional agricultural conditions, without the exploitation that current captivity and factory industrial farming requires. These animals might be even maintained in an environment that simulates their original natural environment. So ultimately, they would be used as a way to source cells from samples that are taken to evaluate the health of these animals that might live in reserves, for example. I believe that the future will be that these animals, or some of these animals, will be maintained not as farm animals, but as wild animals, where with close monitoring for their health, and as a sub-product of that monitoring, you'll have sampling of tissue that could be used to generate new cell samples.
Yeah, so it sounds like kind of in any case, it's going to be so much more ethical for animals. But you know, there's kind of like a range of work that needs to be done there.
I think one of the main drivers of this field is definitely the realization that we are exploiting the animals and treating the animals as a product without any or not much consideration on the ethics and the environment, the well-being of the animal. So that is an important factor driving many of the researchers in this field. Yes.
Yeah, kind of as you said earlier, it's that cultural aspect to it as well. And Judgment Dave says, are you already managing to create artificial meat with the textures and flavors of real meat? And do you include fat and sinew and stuff like that?
We are getting there. But as you can imagine, it does depend, our research does depend on funds. We are very fortunate to have been able to secure funds from the Innovate UK research councils to do some innovation in this area. And we are very much invested in going to the greater challenge, that is really reproducing the texture and taste of the real thing. So recapitulating the precise alignment of the tissue, of the muscle tissue, coexisting with fat cells. So these are projects that we have running at the moment, and they are supported by Innovate UK, as well as the Good Food Institute and New Harvest, from which I am a research fellow. So the short answer is we are getting there. We do have currently some small samples of tissue that are very closely like reproduce very closely the natural structure, composition, and digestibility of natural meat.
Cool. So yeah, thanks. I think we're going to leave it there, but thanks so much for that, Ricardo. The talk was really interesting. Kind of all the questions and your responses to them have all been really great as well. So thanks for doing that. And
Thank you.
No problem. And thanks again to Kyle and Tim earlier for their talk. Also a great talk, really interesting. And thanks to you at home for watching this evening. Remember, you can fill out the feedback form in the description to win some merch. And yeah, thanks again from all of us.